Alrighty, folks, we're going to go ahead and get started. All right, can I have your attention, please? So before we get into class material, I just want to plug again this Cookies and Conversation initiative that I kind of put together earlier in the semester and we're trying for the first time. So this evening is the last one for the semester. This is my colleague and friend Alexandra Naba. She's awesome, a brilliant scientist who works on cancer and developmental cell migration through the extracellular matrix, and a really friendly person to talk to. So if you haven't checked it out yet, if this guy really, wasn't working for you, he's kind of, I don't know, I wouldn't go see him. But she's great to talk to. So please go talk to her if you'd like to try it out. It's just you show up, you have some cookies or muffins or whatever they have, and chit-chat with a faculty member who you get to know. So please check it out if you haven't had a chance to yet, if you're interested. No pressure, obviously. Okay. With that, we'll pick up where we left off. So today is going to be a little bit of a mix of topics, right? Because, again, fire alarm stuff, we're running behind. So this was supposed to be finished already, but I'm going to finish off cancer today by, if only we could actually finish off cancer, right? I'm going to finish off talking about cancer by covering the topic of metastasis. And then I will talk a little bit about a type of cell called a cancer stem cell, which is really important for you to know about. And that will be our segue into stem cells. So then we will cover stem cells for the second part of this class, and then a little bit of the class on Tuesday, because I want to end with at least half of Tuesday's class, if not more, available for a review of the semester. And for that, your TAs, there are, for each of your sections, so for Unit 3 and 4, and I think Dr. Lin's done something similar, if you go to Unit 3, 4 folders, you're going to find review slides that are just, highlights of the things we've talked about, just to help you remember some of the topics to cover. Those are in there as our fake homework questions. And so what we're going to do on Tuesday, the second half of class, is your TAs will take turns going through the Unit 1, 2, 3, 4 material in some small, whatever time we have, with less of an emphasis on Unit 4 because we just covered it, right? We want to remind you of the earlier things you need to know for your final exam. And of course, I'll be here, and I think Dr. Lin will be here too to help answer any questions. And then I hope you saw I have extended office hours that Tuesday, not just before class like I always do, but after class as well. So please take advantage of these opportunities, your TA's office hours, to ask any remaining questions, cover any remaining things that are on your mind before you are hit with exam 4. And then unfortunately, based on the university scheduling, your final exam is pretty soon after that on, I think, Monday or something. So, and again, we have a class policy. I'm not going to get into it in detail now because it's all written out for you, where your lowest exam is dropped. So hypothetically, if you're really happy after exam 4 with your grade, you don't need to take the final exam, but you must make that determination. I'm not going to make that decision for you. It's pretty easy to do that. I've put it in equation form in the syllabus. The FAQ has a whole list of advice. Just kind of follow directions, do the addition and division, right, your average grade, and see what you think. And on that note, your exam 4 being on Thursday of next week, we are going to do our best to get those grades to you as soon as possible so that you can decide what you want to do for the final exam. No guarantees because we are dependent on the grading office, but the plan is, and the TAs and I will go over this, is as soon as we take that exam on Thursday, we're going to rush it to the scoring center, and the TAs are going to block out time to try and double check and verify grades throughout the day on Thursday so that hopefully by Thursday evening you already have your grade. Worst case Friday morning, but we're going to try to get it to you Thursday evening. That way you have several days to figure out what you need to do with the final exam. Okay, so that covers all the non-scientific stuff I wanted to briefly mention today. So getting back to where we left off, metastasis, right? The idea that to be a cancer, an original tumor in most cases has to spread. It has to then go to a location or locations where it's going to cause problems for you, damage to tissue or inhibition of the function of a tissue. For example, stopping your liver from working properly or stopping your lungs from working properly. This slide I'm not going to cover again today because it's what I covered last time and I stopped at it. But I will show you this next slide which has the exact same information slightly differently. And I do this sometimes, right? I give you a couple of slides, whichever one you like better. And I'll point out a few things on this slide that are not well emphasized here. Okay, so what you're seeing here is a tumor in this pinkish color. And don't worry about these different genes and proteins. We're not going to cover them in this class. They're just part of this figure. You may recognize some of the names because we've talked about a couple things here and there, but don't worry about them. What I want you to take away from this slide is that you have this tumor that forms, you have an accumulation of mutations like we've talked about, and at some low frequency that can lead to cells gaining the ability to migrate away. And these purple cells are cells that are changing their shape, their genetics, rather not their genetics, sorry, their transcriptomics and translotomics, so their expression of RNA and protein. It changes so that they become different cells. That whole process we talked about of a cell changing what it is by changing slowly its regulation of what genes are being turned into RNA, being turned into protein. And then you have these cells that gain the ability, therefore, to migrate out into the tissue, and that's the beginning of metastasis. So that's also what's shown right here, same thing, metastasis beginning, because these cells are invading tissue and migrating. And to do that, this transition, going from a cell that looks like this to a cell that looks like this, is called epithelial mesenchymal transition, something you need to know. It's often abbreviated as EMT, all right? Epithelial mesenchymal EMT transition. The transition, I hope, is self-explanatory. The epithelial mesenchymal means it's going from this to this, and that's really what's happening here. This is called an epithelial cell type, which broadly speaking is a cell that basically sits in place, has a pretty standard shape, and we're going to get into those details in a second, and does its job. To be a mesenchymal cell, you look more like this. You actually have polarity, you're going to migrate, you're going to do something different. You're moving, you're not staying in place. So this transition is important because without it, literally the cells can't move without being mesenchymal. Once you do this movement, then remember we talked briefly about how you can invade a blood vessel, float along like you're in a tube in a lazy river or something. Most of those cells will die happily, but 1 in 1,000 roughly, I believe, is the stat. Yeah, less than 1 in 1,000 will survive to form metastases, so less than 1 in 1,000 will actually escape at some point from the blood vessel. And then again, you have to migrate from having escaped from the blood vessel to a target location. In this case, what's shown here is the lung and here's the bone. So now these cells have to migrate, just like they did here. And then at the end, they have to reverse this process. They have to go back to being epithelial. So the only thing that changes in the name of that is you switch the order. This was an epithelial to mesenchymal transition here, and this is a mesenchymal to epithelial transition here, MET. So EMT and MET, all right, 2 terms you need to know. All right, so that's the basics of metastasis in a very high superficial level. You have to get away, and you have to change your cell type, migrate, get into a blood vessel, go somewhere, come out, migrate, and then transition back to being a cell that's going to take up residence somewhere and make a tumor. So let's talk more about these states. What is an epithelial state? What is a mesenchymal state? So when you have an epithelial state, like I mentioned, this is kind of what you look like. You have a very particular shape, and you're hanging out with a bunch of other cells all on a line. These are some of the characteristics of an epithelial state. You have a polygonal or columnar shape. In other words, you look, for example, here, you look kind of like a rectangle. Now, you don't always have to look like a rectangle, but you look like a very defined shape, okay? It's not something amoeba-like. It's just a very clear-cut shape that serves the function in the tissue that you're in. You have apical-basolateral polarization, which is a very fancy way of saying the cell looks different at its top and bottom. So basolateral, baso for basal, base, the base of something, the bottom. This would be the basal side. Lateral means to the side, so basolateral polarization, as opposed to apical. Apical is the top side of something. All right, so all this means is that you have differences between this side, and maybe including this side. Strong adhesion. Remember we talked about this back here at the end of the last lecture, that if you're stuck to a cell, you're not going anywhere, right? And so before you transition in this stuck state, you have strong cell-cell adhesion. You're stuck here as part of this group. You're not metastasizing. Limited migratory potential. Makes sense based on what I just told you. And these are some of the markers of cells that are epithelial. We're not going to cover all of them. The only one I've mentioned to you is E-cadherin, which I brought up here at the end of this last lecture where I talked about components of being sticky. How do you grab onto other cells? And cadherins are proteins that adhere for adhering or grabbing, sticking to something. These are proteins that are known for grabbing onto other cells and hanging onto them. So that's basically epithelial state. You are structured, you have a shape, you have polarity of a particular type, you're stuck in place, you're not going anywhere, and you express some of these proteins. Mesenchymal state here, which is these cells that are moving out, are very different. They are now spindle-shaped, or kind of like an abema, or if you've seen those videos I've shown you with the bacteria moving, they just kind of, they don't have a defined shape that's consistent, but they're always changing their shape to move, right? It's like silly putty, kind of. So they're spindle-shaped. In other words, they have some longevity to them in one direction. And their polarity, all cells are not all, I think almost all cells have some type of polarity. So in this case, your polarity is anterior-posterior. In other words, the front of the migrating cell and the back of the migrating cell look different. You already know this because we covered in cell migration in detail that polarity, right? We talked about racks at the front of the cell, actin filaments forming and protrusions reaching out. We talked about actomyosin-based contraction at the rear of the cell and pulling it up, and the expression of rho. That's polarity. So you've already learned about the polarity, the anterior-posterior polarity that's a part of a migrating state, cell that's in a mesenchymal state. You have cell-cell contacts. Don't worry about the focal aspect of it, but basically it just means that cells are kind of touching each other to see where to go. You have strong migratory potential. Okay, that's what we're talking about. And these are some of the markers. Now, one thing that can get a little bit confusing, notice there's a cadherin here. I just told you that cadherin is there for cells to stick to each other. So why would a migratory cell have a cadherin? That's because the actual situation is more complicated. There are a bunch of different types of cadherins. Some are used to be really tightly stuck to a cell right next to you, and others are expressed in migratory cells and are used to make cell-cell contacts to just realize that another cell is there. Okay, so it really depends. The way you can kind of remember this difference, because we are talking about cadherins, and I am emphasizing that to you, is that E cadherin and N cadherin. E cadherin for epithelial, E for epithelial, and then N cadherin, you can remember that N is pretty close in the alphabet to M, right? So it's not M cadherin, but close enough. So if you remember that way, E cadherin tends to be in epithelial cells that aren't moving, and N cadherin tends to be in mesenchymal cells, migratory cells, again another M, that are moving. All right? I saw a hand, yeah.
Why mesenchymal cell would like to become epithelial again?
Great question. Why does a mesenchymal cell want to become epithelial again? When you get to this location, you're trying to find a home in a sense. You have to stop migrating. This is part of your programming at the moment. As a mesenchymal cell, as I'm moving. And if you try to take up residence in a location, let's say on a lung, you're not able to stick to anything. You're not able to actually grab on and hang out and get energy and all those kinds. It's harder to do it. So the cell will actually undergo this transition state in response to noticing, in response to signals that, okay, I'm in a location that has a bunch of cells that are more epithelial, I should join them. And it'll change its shape. It will transition so that it can plug into that structure, hang out, and grow. Yeah. Any other questions? Yes?
Does it migrate again?
Wow, great question. Does it migrate again? You mean after it's become epithelial?
Yes.
So theoretically, yes, it has the potential to do that. I don't even think it's known how often that happens. What you're talking about essentially, so let me actually be a little bit stronger on that point. It certainly does happen, but we don't really know with what frequency. Because what you're actually saying is you have a primary tumor, you metastasize, you form a secondary tumor. Can that secondary tumor in turn give rise to more tumors? Yes, absolutely. The spread of cancer does work oftentimes that way. But we don't really understand right now, well, the frequency with which that happens. So yeah. I don't know. Any other questions? Yeah.
So is it usually from the host tumor that it mainly spreads?
Right. That's more well established, that usually the host tumor is metastasizing a lot and that those things disseminate out. But, by definition, the cells that move out have the mutations to do that, and therefore so should the secondary tumor that forms from those cells, unless you get other mutations that get rid of your migratory ability, which is part of the reason it doesn't always happen, because these cells are rapidly mutating, both in ways that help the cells and hurt the cells. But there's no reason to think that those cells couldn't again change their state and migrate out. I just don't. It's not clear how often that happens. Yeah. Yes?
Becoming the other shape and move, does this happen only in tumor cells, or does this happen in other?
Great question. Does this only happen in tumor cells or in other cells? That's a fantastic question because it very definitely happens in other cells. I don't think we covered it in this class. It's more of a, dev bio lecture, or maybe I could try to work it in here one day. But epithelial-mesenchymal transition and mesenchymal-epithelial transition are very fundamental processes that happen when you're young and developing, but also later in life. So, for example, you've heard me mention a stem cell type neural crest cells that migrate like cancer cells. In fact, I think I will come back to them later. We study them in my lab. They move all over and they become stuff to migrate out from your neural tube where they first form when the embryo is developing, and to move out, they undergo EMT. The first thing they do, one of the first things they do, is they undergo epithelial mesenchymal transition, and they actually will change from the E-cadherin expression to the N-cadherin expression. They will change their polarity. They will do all the things that were on this slide to move out. And so that's one of the reasons actually to study some very basic cell migration, basic science, even in bio or other fields, because it can actually inform how cancer cells behave. And one of the major lessons to take away from this section of class is that cancer is co-opting our processes, whether that's the cell proliferation, apoptosis that we talked about, overproliferating and all of that, but also things like taking the cell's ability to migrate, which is important, and using it for cancer to metastasize, undergoing EMT. These are not things that are sort of invented de novo. They are being co-opted by the cancer. So absolutely these processes are present in a lot of your different cells, including, say, an immune system cells that are grown and then have to move out, as another example. Yeah. Yes?
So kind of continue that, would it be accurate to think of this as a different behavior that they're rebelling against? Just like how cell proliferation is
Well, so the question is about, is this an example of cells rebelling against the behavior? Not quite, because in the examples of proliferation and all, et cetera, you are rebelling against the checkpoints, the safety mechanisms, right, that stop you from doing those things. In this case, you're co-opting a process that's there. You're kind of taking advantage of it rather than rebelling against it. Although you could look at it differently. I mean, this is getting a little bit into just how you want to think about it. You could make an argument. You could say it this way. You could say that the epithelial cell is rebelling against being epithelial. It is co-opting an existing process of becoming mesenchymal to rebel against its natural state. So you could think about it that way, if that, yeah. Yes? I love these questions. You guys are awesome. How do they know where to go? A big part of the answer is they don't. So a lot of this is, when I show you something like this, it makes it look like it's like a targeted thing, right? But it's actually not. You've accumulated mutations that make you migratory, so you kind of start going out. And most of those guys will die. Once in a while, you hit a blood vessel. Most of those men aren't going to be able to get in. Once in a while, one gets in. Most of those are going to die in the blood. Once in a while, one gets out. Most of those are going to die in the new location. You see? So it's like this, it's this selection that happens with rarity for the few that manage to get somewhere. And then if you remember, we talked about last time, the environment you get to, the seed and soil hypothesis, the Goldilocks idea, you have to have the right home. If you make it through this whole gauntlet, then there's still a good chance you're going to get to a lung or a liver or something, and you're going to die there because you don't have the right pH or the right, signals telling you to stay alive or whatever the case may be. So they don't really know where they're going, as far as we know. It's just that they are disseminating. Now, at a very specific molecular level, they do respond oftentimes to signals that tell cells to move towards the signal. So you can have chemotaxis where you have a ligand, like that example in the test question about the chewing gum, and where you have a ligand that attracts cells towards the ligand. And if a cancer cell is overexpressing a receptor that responds to it, it'll go that way as a direct response to it. But it's not like it knows where to go. It's just responding to signals that may or may not take it to a place where it survives. So there's a lot of randomness here, for sure. Yeah. Oh, yes. Go ahead. How do the cancer cells enter the bloodstream? So I'll give you the short answer because, we don't have time. Remember I told you they're secreting matrix metalloproteinases that help them get through stuff? A subset of them, or many of them, can secrete ones that will also start to degrade the basal lamina of the vessel and start to chew their way through that. There's active research on this. There's a wonderful researcher in C. elegans, Worms, who was here a few weeks ago, who gave a lecture about this, where he actually looked at how much of that movement through a layer like this of cells breaking through is an enzyme chewing it versus physical force. So it turns out, actually, in some cases, Cells will send their protrusions, those lamellipodia we talked about and filopodia, through little openings, and then they can actually put more material in and squeeze the opening wider. Yeah. And there's also a really cool paper that came out. I'm completely off topic, but now you're getting me excited about it. So there's sorry, that didn't sound right. There's times where there's a paper recently that showed that cells can actually, in some cases, use their nucleus. So let's say you made your way partly through a hole. You can take your nucleus and kind of push it forward and use your nucleus as a battering ram to actually go through. And then once that happens, other parts of the protrusions will retract themselves in response, like, oh, okay, the nucleus found a way. We're not going to try and get through those holes. We're going to go through this hole. And this is active research. I mean, like, there's a lot of stuff we're still figuring out. It's a cool topic. Yeah, there's another hand.
Can they go towards the more energy source, like the?
Can they go towards more of an energy source? Sometimes, yes. And so the way I'll answer that is to think about the presence of blood vessels that provide that energy, right? As well as nutrients and oxygen. So cells that are metastasizing, they can detect, let's say, factors that suggest the presence of a blood vessel, and maybe that encourages them to go in that direction. But again, it's random chance that they evolved the ability to do that. Yeah. Any other questions? Yes.
What makes them transition?
What makes them transition? That's a hard question. What makes them transition, I mean, so, you know, we talk about this accumulation of mutations, 47 mutations average in glioblastoma. Some combination of those mutations can trigger these transitions, is the simple way to think about it. It's kind of like, I would look at it this way. If I had a piece of DNA that I can mutate and get 10 different colors out of those mutations, this one makes it red, this one makes something purple or orange or whatever, this outcome of being able to transition is one of those colors. You know, it's like one of the outcomes. It's not that every cell is doing it. Maybe another cell has a mutation that kills it. Another cell has a mutation that makes it more structurally looking like it's just a rectangle. You know, so it's just one of those outcomes. I know that's not the most satisfying answer, and there's probably a lot more to it, but yeah. All right. So, okay, we'll move along. All right. So this slide is I'm showing you EMT in 3 different ways. This one, this one, this one. Again, don't memorize all this stuff. I have one reason and one reason only for showing you this slide. It does the best job of showing you that this is not a on-off switch, as in like, oh, 1 second you're epithelial, the next minute you're mesenchymal, which is what this slide makes it look like and this slide makes it look like. But in fact, although this one at least has like cells that are transitioning right there. It's called transition for a reason, right? Like most things we've learned that require new protein, it can be an hours or even days long process to become a different cell. And so what this slide shows you really nicely is that this very epithelial cell with some cilia over here, and it's very rectangular, and it's part of this nice row, it actually goes through a very gradual progressive loss of these epithelial cell markers that are shown here that we don't need to memorize, and gaining these mesenchymal markers, as well as the changes that are described right here, right? And it just takes time, and then you have a mesenchymal cell. So that's the only takeaway here, is the gradual process, hence the term transition. Okay, so what happens to these cells? We've already covered a lot of this, so I'll be really quick about it, but traveling cells are vulnerable to attack from the immune system, so that's why most of them die. They must be able to attach and grow in some new tissue. They must avoid the immune system and find that fertile ground that's the right home for them. And the mechanisms which allow them to do this are not fully understood, right? So I've tried to give you a flavor of what they're up to. We're limited, of course, by class time and my own knowledge, but we're also limited by what we as human beings understand. This is an active area of research. Metastasis is what kills, so it'd be great if we could figure out ways to stop it. These are some of the examples of things we've talked about. Evolution at the cellular level, variability of the genes. The more you divide, the more you have mutations. Selection of the mutations that could help you metastasize. And of course, the proliferation of cells, as we've covered. Cancer cells can change so much that the immune system no longer recognizes them as self. That can be a good thing because then they can attack those cells. So this is an electron microscopy image with a cancer cell in gray, and these are immune system cells in green. These are false colored just so you can see it better. And these immune system cells are attacking this cancer cell because it's changed so much, they're like, yeah, that's not one of us. That's not a normal cell. And for scale, here's a red blood cell, and you can see how much bigger and really just kind of unhappy looking the cell is that they're attacking. All right? So here then, to summarize many of the things we've talked about, this is another summary slide in addition to the one I had at the beginning of cancer that you can use to help you study. These are some of the things we've discussed. Oh, yeah.
I was just wondering quickly if benign tumors stop proliferating?
Yes. Do benign tumors stop proliferating? Absolutely. You run out of sort of growth potential because you're getting to be so big that you don't have an energy, you don't have enough oxygen getting to you, or a number of other factors. So yes, in most cases, well, yeah, I think in most cases benign tumors eventually stop proliferating, but I'm not super sure about those percentages. Yeah. Any other questions? Okay. So the hallmarks of cancer that we have covered in general include that to grow as a cancer, you have to have self-sufficiency, right? In other words, you're proliferating even when you're not told to proliferate. You don't need growth factors. Those can help, and sometimes they encourage that proliferation, but you mutated in a way that you're able to grow even when you're not supposed to. And one of the examples that we keep coming back to in this class is RAS, right? Remember that oncogene that was on your exam 3 as a gene or protein that if it's always on, you have cell proliferation. So it turns out that 15 to 30% of human cancers have mutant RAS. Now, that doesn't mean that all human cancers have RAS mutated, right? Because obviously most don't. But it's important to note how valuable this is for a cancer, that if you can be always on, even if you aren't supposed to be, then you can continue to proliferate. Insensitivity to anti-growth signals, kind of the opposite side of the coin, but with the same end result. I'm going to grow even when I'm not supposed to, and I'm going to ignore signals that tell me don't grow. Right? So in most cells, mitosis is automatically inhibited because of all the cell cycle checkpoints we talked about. But in cancer cells, these mechanisms are broken. Mutations have allowed these cancers to get around these limitations. Evading apoptosis, the ability to avoid being killed off when you're not doing the right thing. P53 is the most common gene we talked about. It's mutated in about half of all cancers. And again, that means in about half of all cancers, it's not mutated. So don't get the wrong idea that P53 is everything in cancer. It's just that it's a very commonly mutated gene that, and this is a tumor suppressor, because you need to get rid of both copies to get rid of its function so that it can't properly regulate apoptosis. Limitless replicative potential. Tumor cells, they become more like stem cells, and this will be part of our transition into that topic in this class and the next. There's actually some really fascinating similarities between tumor cells and stem cells. They can be quite immortal and relatively undifferentiated. In other words, they keep dividing and dividing and dividing, which is not what your normal cells do. If you have a cell in your muscle, it's not dividing anymore most of the time. If you have a cell in your skin, it's barely dividing, or your top layers are not dividing. These cells, on the other hand, are just dividing like crazy. They're more like stem cells that way. Sustained angiogenesis, you must have a blood supply to grow really big. So, you have to be able to have blood nearby, and we talked about VEGF and BDNF and FGF as examples of that stimulate that growth. Tissue invasion and metastasis, we just talked about that. Decreased cell-cell adhesion, increased motility, making proteases, and E-cadherin being lost or degraded, and then you can upregulate N-cadherin. So, big picture, there have been a lot of big picture slides, but here's one more. Cancer progression is really an example of evolution by natural selection. Remember that video I showed you of those bacteria growing with increasing concentrations of antibiotic? They evolved the ability with multiple mutations to avoid the antibiotics. Similarly, cancer cells dividing all the time accumulate mutations that allow them to evolve and to avoid the mechanisms that would normally kill them off. So it's natural selection. And of course, cells that are mutated and dividing more often will end up sending those mutations to more of their progeny. So that has implications for treatment. You have to target treatment in the right time and place. The earlier you can catch these things. How do we treat multiple mutations? If I target one pathway and I inhibit it, but there's 7 pathways that are mutated, what do you do about the other 6? So there's a lot of important reasons to understand that this is a phenomenon of evolution. Many cancers are linked to environmental factors, suggesting that many cancers could be avoided. So, this little cartoon goes back to my my screed about not smoking, which is by far the most important behavior you can undertake to reduce your chances and your family's chances of cancer from secondhand smoke. It's really, really bad for you. I'm sure you know that by now. Here are some graphs that I had mentioned earlier that I would get to eventually. Most cancers are linked to smoking. By the way, it's not just your lungs. You are introducing toxins that mutate DNA in a variety of cells, and those toxins and those mutations can happen outside of your lungs as well. Also, lung cancer metastasizes, so you end up sending it elsewhere. Epidemiology predicts that if no one smoked, total cancer rates would probably be about 1/2 of current levels. All right, so even though we've done a great job of treating cancers better, detecting them better, we also have behaviors that counteract some of those successes. Here is a graph that I'd love to show. Look at the change in cigarette consumption from 1900 to 1980. This is in men, and we'll get to a comparison between men and women in a second. But if you look at this, you have cigarette consumption that rapidly increased, and this is at a time, of course, when men tended to leave the house, and so they were smoking more, and that's why this graph is about men, because you had this massive explosion in smoking taking off, and then about 20 years later, you see a really nice correlation in increases in lung cancer. And then it started to fall off eventually, if you were to continue this, when people started to get better about that. Notice this graph. It's this information, but with multiple cancers. So here's lung and bronchus in yellow. This is what I just showed you. And then this is colorectal cancer in purple, stomach cancer in gray, and breast cancer in red. Notice how much this increased, while the other ones actually didn't increase. In fact, they decreased, in part due to treatments, but also in part to something we've talked about, the idea that we all have to pass away from something. And if a bunch of people are dying of lung cancer, there's less people left to die of the other cancers. Sometimes it's really that simple. So there's a lot of reasons for that, but while we've made progress in treating many of these, look at this remarkable increase that happened, right? Oh, and I forgot to mention, this one is females and this one is males. So now we're talking about males and females. And this is a fascinating graph because look at the men again, how rapidly it increased here and the decrease in other cancers over this time period. This rapid growth for women happened much later, so about 1960, 1970. What happened? Women's liberation movement, right? Really a good thing, obviously. But the side effects of good things, women got out of the house, they were going to bars, they were going to workplaces, people were smoking all over the place, including on airplanes. And so all of a sudden, women who had been relatively protected from high incidence of lung cancer started to get it 20 years later, and to eventually get up to levels that the men were at a few decades before that. So, don't smoke. Just don't smoke. Okay. Increased understanding of cancer is leading to new treatments. This is just an FYI slide for fun. We've talked about a few of these things, VEGF signaling. Some of these we haven't talked about. But the idea is that by targeting different processes that are dependent on different signaling pathways, we can take a multi-pronged, multifactorial approach to trying to fight cancer by inhibiting different things that cancer needs to survive. So with that, I'm going to start to transition into stem cells by first telling you about cancer stem cells. And I will start that by telling you that cancer stem cells, the name is confusing, okay? And this is not in your book, but I want you to learn it because you will go to workplaces, including being a physician or a nurse or a researcher or what have you, and you're going to hear about these. It's a hot topic right now. So your book is outdated, and I want to teach you what's important. This is important. All right, so what's a cancer stem cell and why is it not a regular stem cell? We'll get to stem cells in a bit, and stem cells are cells that can become different types of cells, and we'll get to that in more detail. Cancer stem cells are named because they are the stem cells of cancer. That doesn't mean they are the same thing as stem cells in general. It just means they are the stem cells of cancer that do a very specific thing that is similar to what stem cells as a whole are described to do. So let me explain what I mean by that. Let's first look at this side. This is the stochastic. Stochastic means random. Stochastic model of cancer development, where you would have a tumor, and in that tumor you would have cells dividing, as shown here, and you would have the ability of some of these cells to keep renewing themselves and keep dividing. So the stochastic, and then within that group you would also have cells in green here that have differentiated. One of the things that we'll cover when we talk about stem cells that I'll tell you right now, you may already know, is that differentiated cells tend not to divide. once you become a muscle cell or a skin cell, you're not usually dividing. To divide, you usually have to be some type of a stem cell. So in a cancer, which is mutating and doing all these crazy things and proliferating, wouldn't it be nice if all these cells differentiated and then they wouldn't divide anymore? That actually does happen sometimes, where you can have some of these cells that by random mutation or chance or the right environment, they differentiate and then they're not really cancerous anymore. Unfortunately, you have enough cells that self-renew. So in the stochastic model, a tumor is a combination randomly of all these different cells doing different things. The cancer stem cell model doesn't quite see it that way. In the cancer stem cell model, you actually have a group of cancer stem cells, usually in the middle of your tumor, that are rapidly dividing constantly. And then as the cells grow out, cells on the outside tend to be more differentiated. The idea is they're more exposed to environmental factors that they're moving through or that they're around that could cause them to differentiate. And that you have this core population of stem cells, usually again at the middle of the tumor, that keep the tumor alive because they keep it replicating over and over again. So why is this important? Why should you care about this? Here's why. Ignore all the text, although you can read it on your own time if you're interested, but here's why this matters for therapy. Traditional cancer therapies, so here's a tumor, and this yellow thing represents a big group of cancer stem cells, CSCs in the middle, and then this is the rest of the tumor. With traditional therapy, let's say you put in a chemical or inhibitor or something, by definition you're going to reach the cells on the outside first and most easily. And as you go deeper into the tissue, your effective concentration of whatever you added is going to go down until it may not even be that effective. So what happens is that you're doing your therapy, you put your drug on there, you're going to kill a lot of these outside cells, but oftentimes you won't kill the cancer stem cells. So you've done this therapy, you're really excited, you shrunk your tumor, great, go back home patient, let's see what happens. 6 months later, the tumor's grown back. Because these cells were still there, and these are the ones that unfortunately tend to divide. I should say not grow. They tend to divide, which makes the tumor grow. So the hope, the idea, the reason this is important is that can we get to a point now, and this is a very new idea. It's maybe 10-ish years ago that it started to really catch hold, that tumors have a core population that we should be targeting. And that's difficult to do. Now that that idea has gained traction and most people accept it, it's changing the way we think about cancer therapy, so that you want to actually devise drugs that will kill these stem cells, not even worrying about these other ones because they're fairly differentiated. If you can get to the core, if this is the Death Star, and you can get to the middle of the Death Star and destroy it from the inside, if any Star Wars fans are here, then you've destroyed the whole Death Star, and you don't need to worry about any pieces that fly off of it. So that's the idea, is that can you target the cancer stem cells, and there's a lot of active research on that. So I want to share with you one example of that active research. This is a really cool paper that came out a couple of years ago. And again, this is just for fun, this part. So I want you to know what cancer stem cells are, but I'm not expecting you to know all this for an exam. I just think it's important to share. You may remember the Zika virus, right? It was all in the news a couple of years back. Dangerous virus that infected people, could be dangerous for kids, in particular if the moms were pregnant. This group put out a really interesting paper. They found that this virus actually has oncolytic activity. In other words, it's able to destroy cancer cells, preferentially glioblastoma cancer stem cells. So I'm not going to read you the whole virus, but they kind of tried a few things and found that if you took this virus and put it on a bunch of cancer cells, it tended to actually kill the glioblastoma, which is a very deadly cancer. It tended to kill those stem cells far more than it killed those outside cells. So how exciting is that as a therapeutic opportunity? You're worrying about all the time being able to get this group. What if you suddenly find a virus that actually targets this group? All right, so there's a lot more work to do, but this is a really exciting, one example of an exciting opportunity of trying to target cancer stem cells using things that we've co-opted from nature, which, by the way, is the vast majority of what we do in scientific research. We are constantly taking things from nature and reusing them for different means. CRISPR-Cas9, for example, that you heard about from Srivastava, was something that bacteria used to kill viruses, and we've now used it as a gene editing tool. So this happens all the time. Kudos to these guys for finding this. And hopefully it leads to something interesting down the road. Okay, let's see, it's 11:40. This part is not going to be on your exam, okay? So if, you can kind of relax. I'm going to share with you a little bit more explicitly. I've alluded many times to the research in my own lab. All right, so this is a good time to share a little bit of it, some unpublished work, and just to give you an idea of what we actually do. And I'm going to go through this fairly quickly for time, but gives you a little bit of a sense of what we're up to. Neuroblastoma is the most common tumor in the first year of life. So kids can get this, and that's, that's tough, right? I mean, I'm a cynic about adults personally, but I love kids, right? I think you can really shape young minds, and it's always tough to watch a young child dealing with something like this. So I'm motivated, my lab is motivated to understand this cancer and how we could try to fight it. And we do very basic science research to understand that. So this is a tumor that starts in very early forms of nerve cells found in embryos or fetuses. Neuro for nerves, blastoma, a cancer that affects immature or developing cells. Here's what's really interesting about this. Sometimes in very young children, these cells die for no reason and the tumor goes away on its own, and that's not well understood. So if we know a tumor can go away on its own, what if we can understand it? And if we can understand it, what if we can make more of those cells go away? That's kind of the goal. So I'll skip through some of this. I'm going to show you something. It's a video. I'm showing you this video for two reasons. It actually shows the work we do and why we do it in zebrafish embryos. But the other reason I want to show it to you is that this was made right here at UIC by a biomedical visualization student, graduate student, Veronica Zokler, who now has a job in Colorado. And she collaborated with my lab to make this. So it's a really neat example of what somebody at UIC can do, and it's really high quality video that she made. So I'll just play it for you guys for fun. Oh, sorry, I forgot to unmute. Let me start that over. Okay.
Neural crest cells are multipotent stem cells derived from the embryonic ectoderm. These stem cells contribute to many structures in the embryo, including the nervous system. Neural crest-derived cells can sometimes give rise to cancer, such as neuroblastoma. These tumor cells share similar gene expression and migratory behavior with neural crest stem cells. Stem cell migration can be studied in zebrafish embryos, which share many of the same genes that influence human development. Cranial neural crest cells migrate to specific streams. When injected in zebrafish embryos, neuroblastoma tumor cells migrate to similar streams. The study of these stem cells and tumor cells could provide insights into cancer metastasis and new targets for cancer treatment.
Monica made this, which is pretty cool. I mean, she's good at what she does, that's for sure. Yeah. Okay, so that gives you some sense of what we do. All right. Yes?
Checking the neural tube in a fetus. Are they able to detect anything like that through ultrasound, or do they have to take blood samples or anything like that?
So the question is, in an 11 or 12-week-old embryo, when you're doing ultrasound to look at if the neural tube is forming correctly, can you detect something like this, a neuroblastoma forming? Unfortunately, no. It would be very difficult to detect it at that point, and I'm not even sure if the cancer is evident at that point. So no, not at that point. Yeah. Right.
But typically, how long, after birth or something, is it diagnosed?
It depends. Sometimes it's detected pretty soon after birth. Sometimes it's later in childhood. 1 year, 2 years, 7 years. It really depends. There's different types of neuroblastoma, and it's a little bit more complicated. There's something like 30 some odd subtypes. Some are more malignant than others. Some are more metastatic than others. Yeah, but it tends to be detected because somebody's like, okay, my kid's complaining of the following symptoms. They go to the doctor. You go through the screening process, and you discover that you have this. Unfortunately, yeah. We know it can take hold in utero, in the embryo, because of research. In other words, people have asked the question, well, gosh, we see it so young. When does it first start? And so people have looked backwards and been able to detect it that way. But it's hard to see it if you're not, doing that type of, All right, because it's getting a little bit late, I'm going to, I want to get started on stem cells so that next class we have time for your review, right? So I'm going to skip this stuff. If you're interested, you can kind of watch or read about this, or, I've told students you're welcome to contact me and come see our lab. We do lots of tours for people. So blah, blah, blah. Okay. I will show you a cool video real quickly. These are neural crest cells in green migrating out from the embryo. This is a time-lapse video, and these are cancer cells in red that actually follow the neural crest cells.
I think that guy that was here like April 4, he showed us something.
What guy? Sorry?
When you didn't teach the lecture, you just telling us about like the scans and stuff.
Oh, he showed you this?
Yeah.
Oh, he did? Okay, I didn't.
He showed
Okay.
several of those things.
Yeah. So Srivatsan's a grad student in my lab, and he's the second author on this paper that we're going through the process with. So I didn't know he showed you that. That's great. Okay. All right. So we will skip all this fun stuff. I will show you this one. I showed it to you before once. Well, one of the things we're able to do is we've identified regions of the embryo where you can put in these cancer cells. These are human cancer cells, neuroblastoma cells, and cause them to become neurons. So you're going to see this cell take on the morphology of a neuron, and we can do antibody stains and show they're neurons, and then they die. They undergo apoptosis. So that's kind of one of the things we're working towards. Remember I told you that these cells sometimes can on their own in human patients go away. And so we think part of why they go away is that in the right environment they can be forced to become neurons that then die. And that's basically the work we're excited about that roughly half of my lab is doing. So yeah, that's a bit of a flavor of that. Okay, so with that introduction then I'm going to now talk to you about stem cells, and then we'll continue that next lecture. Any last questions about cancer before I move on to that topic? You guys have been great about asking really fun questions, and I hope it's been useful for you. It's been fantastic for me. I really don't like talking for an hour and 15, so it's really fun to be able to answer your questions. It's much appreciated. Okay. So stem cells. These are the learning objectives and questions to answer. And, it's regretful that your exam is going to be pretty soon. Right? We're going to start today, then we're going to finish it Tuesday, and you're going to have to know it for Thursday. But you'll notice immediately that the topics, because of that, are fairly straightforward. I'm not going to try and ambush you with a bunch of stuff right before exam 4. These are the things you need to know for that exam, the learning objectives, the questions to answer. What are stem cells, et cetera? What does development mean to you? Differentiation. We'll talk about extrinsic versus intrinsic signals, morphogens, stem cell therapy, and perhaps most sinisterly, am I your original professor or a clone, and will you ever know? Some questions are best left unanswered. So this is, in one slide, stem cells and why they're so important. How do you go from a single cell to a well-formed creature, an animal, a plant, whatever, that is so different from the next thing that came from a similar cell? So if you look at these here, there's a difference in size, but if you ignore this outer sort of coating that holds it in place, same thing on here, and look at this cell and this cell, they don't look very different, do they? They're really similar. So how on earth do you get to something so different? It's basically a lot of what we've talked about, the ability to turn various genes on and off, make different RNA, different proteins, make different decisions about what a cell should be. It is genetics and cell biology. That's what it is. So let's talk about what stem cells are. Stem cells, that term is often confusing, particularly because it's so commonly talked about in the media, and people think that stem cells are cells that can become anything. That's not true. Some stem cells can become everything, most cannot. All right, so you can impress people by telling them about what I'm going to tell you about, because they'll be excited that you know what a stem cell actually is. Stem cells are a special cell type that remains relatively undifferentiated and unspecialized, and can thus give rise to multiple cell lineages. Notice how vague that is. Oh, it's a special special cell type that's relatively undifferentiated, relatively unspecialized, and can give rise to multiple lineages. Not all lineages, but multiple. So to put it more simply, this is really the take-home message for stem cells. A stem cell is any cell that can make itself and or make other cell types. That's it. So if somebody asks you in one sentence, what's a stem cell? A stem cell can make more of itself, and it can make other things. That's it. So those other things could be one other thing, two other things. It doesn't really matter. It counts as a stem cell. Because the important point is that it can differentiate in all these different some of these different cell types but it can also make more of itself, so it doesn't lose itself. You know, if you had a stem cell that just made 2 muscle cells or 1 muscle cell, that's it. You don't have a stem cell anymore. But if you have a stem cell that says, I'm going to make 1 more stem cell from me, and I'm going to make a muscle cell from me, then that stem cell it made is able to keep being a stem cell. So that's why the replication is so important, to continue the process of being a stem cell. Yes?
Is it possible to grow a whole organ out of one stem cell?
Is it possible to grow a whole organ out of 1 stem cell? Yes, and the more detailed answer to that is, inside of you, that's exactly what happens. In most cases, a organ is probably starting from a single cell that came from other cells. In vitro, in other words, in the lab or something, it's a lot harder to pull that off because you have to get it just right. And that's a whole other topic. From making the right cells, so putting them in the right place, to building the scaffold, right? So like, for example, your lungs have a very beautiful architecture. How do you replicate that in a lab? We are going to get there. There's rapid research on that topic, but it's going to be a few more decades to be able to do that. Yeah?
Are egg cells stem cells?
Are egg cells stem cells? Yes, they are. So those are germ cells, and they are stem cells because you can make more eggs, and then when you combine with a sperm, you can go ahead and make a bunch of stuff. Yeah, but we are going to talk about somatic stem cells. So once you have a zygote, once you have a sperm and an egg come together, and you have that single cell, that's also a stem cell, and then what comes from there. So, yeah. I'll play a quick video for you about stem cells.
All living organisms begin from a single cell. This cell, the egg, divides and therefore multiplies. While multiplying, the embryonic cells are specializing. Some will become neurons, others blood cells, resulting in more than 100 different types of cells. So from undifferentiated cells, we get specialized cells. The undifferentiated cells are called stem cells because they give rise to so many others. Either all those that exist when referring to the very first embryonic cell, or a mere dozen when the stem cell comes from the already differentiated tissue of a fetus, for example. Stem cells are also present in adult organisms. They are responsible for regenerating our skin and bones. In general, they are only able to differentiate into very limited types of cells. The therapeutic use of such stem cells could be marvelous. Instead of a graft, for example, it would be enough to transplant stem cells, which would then replace those that were missing or deficient. If, on top of that, the genomes of those cells were to be replaced by those of the patient, any risk of rejection would be avoided. Some people are already dreaming of cell banks, or even banks of organs cultivated in vitro, realizing the dream of spare parts for humans. We haven't reached this stage yet, but research is underway, providing massive scope for ethical debate.
Okay, so one of the points raised there was how your skin and your bones, those stem cells can't make a bunch of stuff, right? So that gets to this part of the slide here. The idea that a stem cell doesn't always make everything. So there's 3 major categories of stem cells that you need to know. The first category is totipotent. So potent refers to power. How powerful are you? That's potency. Toti means total or all. So a totipotent cell is all-powerful. It can make all things. The reason I emphasize these prefixes and suffixes is that if you know what they mean, you'll remember the actual definition without having to memorize word by word. Because, for example, here, potent, potent, potent. The only thing that's changing is toti, pluri, and multi. So a totipotent stem cell can make all cells. The only cell in you that can do that is the very first cell to form, the zygote. It can make all cells. That's the only cell in you that can do that. The reason for that is that what happens pretty quickly is, as soon as it starts dividing 2 cells, 4 cells, 8 cells, you end up having now pluripotent stem cells. Pluripotent stem cells are almost totipotent. They can make all the cells in your body. Now you might be thinking, wait, if it can make all the cells in the body, why can't they make all the cells? Because you have an extra embryonic layer, your placenta, right, that you're in. So that's the technical difference there, is that this first cell will make your body and the enclosure for your body, but then this cell will make just the cells in your body, and the one that's not shown here is going to be the one that's going to go off and make your placenta, your extra embryonic tissue. So in the context more broadly, again, totipotent makes all cells. Pluripotent, so like plural, many, can make all the cells in your body. And then you just kind of have to memorize this difference. I'm not really sure of a good way to do it. Maybe this will work. They go in reverse alphabetical order, so it's T to P to M. You get more and more specific. All right, so if you remember that, maybe multipotent can make multiple different cell types, but not all cell types. All right, so let me repeat that all summarized together in one go. Totipotent cells make all cells. Pluripotent cells make all the cells in your body, and multipotent cells make more than one cell type, but not all cells. Okay? Simple definitions if you think about it. So here, for example, a blood stem cell is a multipotent stem cell that can make different types of blood cells. A muscle stem cell is multipotent, it can make different types of muscle cells, and so on. So your organs in vivo, inside of you, come from these stem cell populations that are specific to different regions or different organs or different cell types. And those are multipotent stem cells. The neural crest cells I told you about, another example of multipotent stem cells. They make multiple types, but not all types. All right. So this is a simpler slide, just summarizing the main point about self-renewal. Stem cells are cells, again, to reemphasize, that can become themselves in this process, but also become other types of cells. You will see these terms throughout the rest of your career. Precursor cell, differentiated cell. So it's important to understand that. A precursor cell, pre means before. A precursor cell is a cell that comes before another cell. That's all it means. So a stem cell is a precursor cell. Like, precursors is a broad category. And any cell that makes another cell is a precursor cell. So in this case, proliferating precursor cells just means that you're dividing a bunch of cells that are going to become something else. Terminal means the last or final. Terminally differentiated cells are cells that have differentiated into a type and they can't escape that type. So now you've made your final neuron or muscle cell, myocyte, or epithelial cell in your skin or something like that, and that's your fate now, and it's literally fatalistic, right? You've made that type of cell and that's it for you, unless you try to manipulate it somehow. So let's watch another video.
Such as red blood cells, neuron
Sorry, let me turn that volume down.
cells can differentiate into any cell type in the body, such as red blood cells, neurons, or muscle cells. When grown in culture and exposed to an appropriate cocktail of signaling molecules, previously homogeneous undifferentiated embryonic stem cells organize into groups of highly specialized cells. Remarkably, the cells in these groups start contracting rhythmically and in synchrony, indicating that they have formed a fully functional contractile apparatus and connecting junctions, characteristic of muscle cells. GFP, expressed from a heart muscle-specific promoter, shows that the appropriate gene expression programs are selectively activated in the beating cells.
Okay, so now we're going to go through some bullet points, whatever we have time for today before you guys get out. We still have some time in class. So tissues then are organized mixtures of many cell types. That's what stem cells are doing. They're making lots of different cell types, right? Here's a very simple diagram. Well, I shouldn't say simple. It's kind of unnecessarily complicated. But it's a diagram from your book that illustrates that point. So here you have your skin, your epidermis, with various layers below that. Don't worry about the details, but notice that there are layers. There are layers to this tissue. Those layers look different from each other because they have different cell types. And they have different different cell types because different stem cells made those different cell types. So nerves, the blood vessel, all this comes from different stem cells that are doing different things. If you looked in your epidermis, you would find even more different cell types. Within your epidermis, you have pigment cells that give you color. You have keratinocytes that give you structure. You have an epidermis over here. You have immune system cells. And as you went through each layer, if you looked at it, again, the point is that you have so many different cells in a given layer that are doing different jobs together. That's basically the definition of multicellularity. We function as complex organisms because we've got so many different cell types doing all these different things in coordination with each other. Different tissues are renewed at different rates. Stem cells generate a continuous supply of terminally differentiated cells. Not all cells, not all tissues grow the same way. Some tissues in adults, like your skin, have rapid proliferation. Others, like your brain, have almost no proliferation. So you have different tissues renewing at very different rates. Your skin is an easy example of rapid cell proliferation. As we've talked about, this is why skin cancer can be quite common, because it's a rapidly proliferating tissue. So any mutations that you get from UV light, for example, wear your sunscreen, can put you at risk, because those mutations can go to these stem cells that are down here. So the way your skin works is that your top layer is constantly being shed. I think I mentioned this in class before because I think it's a fun, gross fact to share. Most of the dust in your home is made up of you and your pets and your kids and your family. Right? Your dust is mostly composed of bits of your cells that have fallen off and formed dust particles when they cluster together. So that's kind of fun to think about. And so you have this constant shedding, and that's nice because this is your protective layer, and if you have damage to it or something happens, no big deal. You've got more cells coming up to take its place. And as you dive deep into your skin, you see different layers of different cell types, and down here, you'll see cells being born. These are dividing basal cells. These are stem cells that are dividing, and as they divide, they'll move cells, the cells will get pushed up until they are here as more differentiated cells. And if you look at a section of tissue here, you can actually see this is probably mouse tissue. You have dead cells at the top, you have these layers, and as you go down here, you start to find a lot of proliferation of stem cells. This is another example of highly proliferative tissue, your gut. Again, this makes sense. You're eating food, you have lots of acids in there that are digesting it, you've got lots of enzymes, you have to protect against bacteria. It's a layer that has to deal with a lot of caustic, corrosive inputs. So it makes sense to keep shedding that layer as those cells wear out or get damaged. So this is in your intestine, and this is the layer of the intestine. Notice how it's all sort of, there's a lot of structure here that gives it a lot of surface area, which is important for absorbing nutrients and digesting food and all that kind of stuff. So what happens is that the outside of this is composed of mucus-secreting cells that are doing a lot of important things. You have brush border cells that are moving things along. And then when you go down into here, you have what's called a crypt, and in here you'll find stem cell populations. So if we take this tissue and we look at it in a cross-section of this crypt area here, well, first let me point out to you that there's epithelial cell migration from birth down here to the top up here, about 3 to 6 days in humans. So that means that a week from now, the cells that are going to be over here are entirely different than the cells that are there right now. That's how rapid this turnover is throughout the whole lifespan for you. All right, constant renewal. That's a lot of cell division. Okay, so if you looked in this crypt, what you would find is that in the crypt, colored differently here, are non-dividing terminally differentiated cells. Those are more towards the surface. They've stopped dividing. They're going to do their job now, and they're going to keep getting pushed up here as the more cells are made down here. And if you went here, you'd find dividing precursor cells that are making more of them, and then you'd find a population of stem cells down here that's not only dividing and making the precursor cells, but it's also making more of itself, so that you have a constantly available stem cell population that can divide, push things up here. They get told to divide some more, but they can only become these differentiated cells, and you have this happening throughout your whole life. So these are a couple of examples of how stem cells do their job in adults, not just in embryos or fetuses. So any questions about any of that? Yeah. What controls the function of the stem cells? That's a big question. I mean, a lot of the control is some of the very, expected answer kind of stuff we've talked about. Different types of RNA and protein being made, genes being activated that are known as stem cell factors, stem cell specific proteins that do the job of keeping something in a stem cell state. That includes suppressing the ability to differentiate into other cells. It includes proliferating rapidly, and it includes probably staying in place to do that. So there's a lot of different control mechanisms. At the end of the day, it all comes back to what DNA you're expressing into RNA and protein. It's a good point to mention that because of these cells being the way they are, very undifferentiated and dividing, you could imagine that if you create mutations, you can have a high risk of cancer, and that is actually often true. Yes? Yeah, go ahead.
When pregnant women are donating their umbilical cord blood,
blood or whatever, are they extracting the stem cells from the umbilical cord itself, or how are they doing that?
Yeah, so the question is if you're if you're having a kid and you take the umbilical cord and you donate that, or you're saving it, right? That's an option that's offered these days. Why are you doing that, or what are you doing with those cells? The idea is that that cord those cells are coming from the embryo, and so you can try to clean up, extract those cells, and freeze them back, and you keep them as a resource in case that child has any downstream issues that perhaps its own cells could be engineered to help with. So these days people are doing it as kind of a safety mechanism because the technology is advancing so quickly that there's the option now at high cost and very rarely, but down the road at lower and lower cost and more frequency of being able to use your own cells to help you with something. Now, you may never need them. You have to pay a lot to store them. A lot of people don't do it, but that's why they're doing it. Yeah. Yeah. Oh boy, yeah. Yes, I've heard about that. So what you're talking about is this idea that some people will mush up these cells and make a pill and then eat the pill. Yeah, I wouldn't do that. I mean, it's not going to hurt you. It's kind of, this is where we get into pseudoscience. Yeah, and unfortunately, when the internet came about, the dissemination of information has been a positive thing, but we've seen negative aspects of that as well. For example, political leaders who don't maintain decorum online, or people of any spectrum of belief, whether far left or far right, who will spread false information, right, for their own ends. Or people who make money by being celebrities that say, you should do this thing because I said it's good for you. Those are the negative sides of that internet. And one of the fads that's out there is taking these types of tissues, mushing them up, drying them out, and making pills out of them and eating them. Now, all you're doing is taking a bunch of protein and taking a protein pill, as well as lipids or whatever's in there. It's just calories. I mean, it's not going to, that's about it. I mean, it's a little bit, whatever, right? And that's all I'll say about that. Any other questions? Yes. Yeah. Say that again? I'm not sure. So epi means, what does epi mean? Above, thank you. Yeah, so so it's kind of that top layer above, yeah. Right, basal is at the base, yeah. Totipotent? Well, the only example I'm sharing you with really is, in most cases, the only example is that very first zygote, because it's the all-powerful cell that can become both your embryo and the extra-embryonic tissue. Okay. Any other questions? Yes. Sorry, I can't hear you. How are stem cells what? One more time.
Stem cells are used to kill sickle cells. How are they using it?
They used to kill cells? Oh, sickle cell. Oh, how are stem cells used in treating sickle cell anemia? Is that okay? All right. How are stem cells used in treating sickle cell anemia? So sickle cell anemia has to do with cells being shaped in a funny way, and that tends to create problems with circulation. And I'm not sure, the short answer is I don't really know the answer to your question, but broadly speaking, the idea is, can you take stem cells and correct defects that might be there and then reintroduce them? So, for example, if you have a problem with cells in the blood, can you introduce a stem cell population that you've corrected that problem in, and if those stem cells that you reintroduce into the patient that you took them from can take up residence in the area where they proliferate and make more normal cells that are correctly shaped, then perhaps you can help solve that problem. I don't know anything about the status of that type of a treatment for sickle cell anemia. I don't know how successful it's been, but that's the general idea. Yeah. Yes?
So cancer stem cell develops from mutations in normal stem cell, right?
Does a cancer stem cell develop from mutations in a normal stem cell? It can, but it's not necessarily that way. That's why I'm glad you asked that question, because that's where the confusion can get in, because they're called stem cells, right? Cancer stem cells are called stem cells because of what they do, not necessarily because of where they came from. They're called cancer stem cells because they can make more of the tumor, and they replicate. So they have that same behavior. They could have come from regular stem cells because regular stem cells are more vulnerable to becoming cancerous because of the way they are. but they don't necessarily have to come from there. Yeah. Okay. So the next topic, in brief, will be that specific signals maintain stem cell populations. And at this stage, that's probably not a surprise, right? We've been talking about signals doing a lot of things. Maintenance of stem cells, it makes sense that you'd have signals that help that happen. So I skipped the Wnt signaling pathway for time in the last lecture. So don't worry about what it is necessarily. But this gut is a great example. This is that crypt that I was just showing you back here. And then these are the more differentiated cells up here. And this is just an example of one pathway that can be different in different areas that helps with that. So, you know, somebody asked what makes a stem cell be a stem cell? And my answer has to do with RNA and protein and all of that and pathways or whatever. But this is part of that too, is signals from the outside that then change that RNA, that protein, or maintain it. So in this case, Wnt signaling, which is involves a secreted ligand called a Wnt ligand, binds to frizzled receptors and causes downstream signaling in the stem cells that encourage them to continue to proliferate and to basically divide, because stem cells need to do that. And then on this portion of the crypt of the gut here, you find that there's no Wnt signaling, or almost no Wnt signaling. So this is just one of the exterior, or external, I should say, mechanisms of regulating where you should have division and where you should not. So with that, let's see if I want to, what comes next? Yeah, I'll give you one more slide and then we'll call it because we're getting close to 12:15. Stem cells can be used to repair lost or damaged tissues. So stem cell differentiation can be steered by exposing cells to appropriate factors. This is one example of that in a broad sense, a pathway from, that keeps cells proliferating or not. Other examples are shown here, that you could take cells from an early embryo that can become different cell types, put them in a dish, embryonic stem cells, and add on different proteins or chemicals that might encourage them to become one cell or another. This is at the heart of stem cell therapy that people are doing research on right now. And we'll talk more about this on Tuesday, the idea that we could use stem cells to try and create cell types and hopefully maybe reintroduce them into the patient at some point. So I'll stop there. Have a nice week, and I'll see you on Tuesday. Hey, what's up? Those